Hello everyone and welcome. In this video we're checking out variant 2 of May June 2017. Before we start, make sure to check out the free notes I have available for you in the comment section down below. Now let's go ahead and start with question number 1. Which organelles are enclosed in a single phospholipid bilayer and contain hydrolytic enzymes? A single phospholipid bilayer, a single membranous organelles containing hydrolytic enzymes is obviously lysosomes. Okay, mitochondria are non-membranous, Golgi body do not contain enzymes, and endocytic vesicles are going to enclose some pathogens or some, materials from the outside which are definitely not enzymes. The DNA of prokaryotes is naked and circular. Which statements describe how the DNA of eukaryotes differs from the prokaryotes? Okay, DNA of eukaryotes is going to be it's going to actually be linear and it's going to have histones attached to it, versus prokaryotes, which are going to have histone-like proteins attached to it and are going to be circular. So it has proteins attached to it. Now, the recently discovered Pandora virus measures 1,000 nanometers in diameter, and Mimi virus has a diameter of 400 nanometers. What can be detected using a light microscope with a maximum resolution of 0.25 micrometers? Now, to be able to compare, you first need to convert from micrometers to nanometers, which to do that you just need to multiply by 1,000, which basically gives you 250 nanometers. So using this light microscope, anything less than 250 nanometers apart won't be able to be seen. Otherwise, anything bigger would be seen just fine. So both the Mimi and the Pandora virus will be seen. What are found in chloroplasts and mitochondria? Both chloroplasts and mitochondria have circular DNA, 70S ribosomes, and mRNA, so 1, 2, and 3. So A. Boiling the bones of boiling the bones and teeth from dead animals can be used to produce a type of glue, and the glue is formed from collagen fibers present in bones and teeth. Which statement describes the changes to collagen that occur when the glue is produced? So the changes in collagen that occur when glue is produced: fibers of collagen become more coiled, fibers of collagen become more flexible, the helices of collagen molecules unwind is exactly what happens. Once you heat the collagen molecule, it's going to break all the hydrogen bonds in it, as well as all the bonds that hold it together, and therefore breaking down the tertiary, quaternary, and secondary structure. So the molecules of the helices of collagen are going to unwind. What describes the primary structure of a protein? The primary structure will not be described by an alpha helix. Alpha helix and beta sheets are both part of the secondary structure. A dipeptide, the bond itself does not describe the primary structure. However, what does describe it is the bead-like structure. It's basically the order and sequence of the amino acids in the polypeptide chain. A globular structure describes the tertiary protein structure. However, the specific and order of amino acids is going to be the answer. The diagram shows 4 monosaccharides with the formula C6H12O6. Which diagram shows alpha glucose? To identify alpha glucose, look at the hydroxyl groups. So these guys. What you're going to be looking for is up, down, up, down. Down, down, up, down. So down, down, up, down. That's B. Up, down is beta glucose. Okay, complete digestion of polysaccharides requires all glycosidic bonds between the monomers to be broken. Amylase only breaks alpha-1,4 glycosidic bonds. Which row shows how completely amylase can digest molecules of cellulose, aminopectin, or amylose? Polysaccharide, cellulose, aminopectin, and amylose. Cellulose has beta-1,4 alpha-glycosidic bonds, therefore it will not be broken down, so a minus would be right. Aminopectin has both alpha-1,4 and 1,6 glycosidic bonds, therefore it won't be broken down as well as amylose, which is only made of 1,4 alpha-glycosidic bonds. And so the answer here should be minus, plus, and then plus for amylose, so it's B. The diagram shows the results of tests on 4 solutions containing biological molecules, which shows the solution that contains only starch and protein. So the solution that contains only starch and protein will only be positive for starch and protein. And Biret tests for proteins, iodine test for starch. Benedict's test, however, tests for reducing sugar. So you're not looking for a positive Benedict's test. And all these tests here are positive. So D should be your answer, because you don't want a positive test for Benedict's, because we're only looking for a solution containing starch and protein. The enzyme invertase catalyzes the breakdown of sucrose to glucose and fructose. 3 different enzyme inhibitors of invertase X, Y, and Z were investigated, and the percentage of inhibition of invertase was measured at different concentrations of inhibitor, and the graph shows the results of the investigation. So you can see that percentage inhibition of all X, Y, and Z is going to increase as inhibitor concentration increases, except for Z, where at a specific concentration, maybe 4, the percentage inhibition is going to remain constant. Which are valid conclusions of these results? The higher the concentration of inhibitor X, the less sucrose is broken down. So the higher the concentration of inhibitor X, the less sucrose is broken down. It's actually quite right. Okay, as you increase the concentration of inhibitor, the enzyme does not do its work anymore, so it does not break down any more sucrose. Okay, the production of glucose and fructose using inhibitor Y is higher than when inhibitor Z is used. Okay, inhibitor Y versus inhibitor Z. Inhibitor Y actually has a greater percentage inhibition compared to inhibitor Z. So the production of glucose and fructose should be lower than when using the inhibitor Z. So this is wrong. The production of glucose and fructose at an inhibitor concentration of 2 arbitrary units is lower than at an inhibitor concentration of 4 arbitrary units for all inhibitors. Okay, for at 2, there should be more inhibition. So there should be the production of at the concentration of 2 should actually be higher at concentration of 2 compared to at concentration of 4, which is obviously going to be greater percentage inhibition, which means that there should be less glucose and fructose being produced. So this statement should be reversed. So it's really going to be 1 only. The following statements are about enzymes. Folding of an enzyme molecule causes the formation of the active site. The shape of the active site changes to enable substrates to bind. Temporary bonds hold the substrate to the active site, and more enzyme-substrate complexes are formed at the optimum temperature. Which of these statements are correct for induced fit hypothesis? Okay. Yes, temporary bonds do hold the substrate to the active site, and the shape of the active site does change. The enzyme is going to undergo a conformational change in shape in order to hold the substrate in its place. Folding of an enzyme molecule causes the formation of the active site. Does not just apply for induced fit hypothesis, or is in any way significant to induced fit hypothesis. And more enzyme substrate complexes are formed at the optimum temperature is quite true. So I will have to choose 2, 3, and 4. How is Michaelis-Menten constant used? Well, to assess the efficiency of an enzyme in catalyzing a reaction, to compare the affinity of enzyme to their substrate, to compare or to find the maximum velocity of an enzyme, or to find the rate at which the substrate is loaded by an enzyme. It's actually to compare the affinity of what's called enzyme for the substrate. How the Km is used? First off, what is the Michaelis-Menten constant? Km is supposed to be the substrate concentration at which half the enzyme, half the maximum activity of the enzyme is reached. So basically, if we draw a graph for the rate of reaction against the substrate concentration, you would usually see something like this, where there will be an increase followed by a plateau. Now the substrate concentration, though, this is over here the V max, the maximum rate of reaction. Okay, now this concentration over here is not what we're looking for. What we're looking for is the substrate concentration at which half the V max is reached. So here we have half the V max. Okay, this concentration is actually the Km. So the higher the Km, the lower the affinity of the enzyme for the substrate. What does this mean? It means basically if you increase the Km, you're increasing the substrate concentration at which half the maximum velocity is reached. So this means that the enzyme is not as attracted to the substrate and requires a whole lot of substrate to be there for it to be able to reach only half the maximum rate of reaction. Versus if the Km was lower, so this means that there will be, we require only a small amount of substrate to reach the maximum rate of reaction. This means that the enzyme is really attracted to the substrate, has a really high affinity to the substrate, and that's it. So this is how we use the Km value. Proteins in the cell surface membranes of human cells and mouse cells were labeled with fluorescent dyes. The human cells were labeled with a red dye, and the mouse cells were labeled with a green dye, and a human cell and a mouse cell were then fused to form a hybrid cell. At first, the different dyes remained separate, and after 40 minutes, the dyes were evenly distributed in the hybrid cell surface membrane. So what does this show? Okay, the protein dyes over here are the ones that moved. Okay, so you have to understand with me that the membrane is a fluid structure. Everything is continuously moving and it's all over the place. So, but what the experiment is aiming to show you is that proteins can move freely throughout the membrane. So it's not about phospholipid molecules, it's about proteins. What explains this observation? All protein molecules in the cell surface membrane are fixed structures within the cell, but phospholipid molecules can move freely between them. That's not what we saw. We saw that proteins moved freely, therefore A is wrong. Groups of proteins and phospholipid molecules in the cell surface membrane are attached to each other and move together. Again, this is not about phospholipid molecules. Only protein molecules in the outer layer of the cell surface membrane can move freely within phospholipid molecules. You cannot really prove that only protein molecules are moving freely between phospholipids which are stuck in their places. You can't really prove this using the experiment. Protein molecules in the outer layer of the cell surface membrane and those which span the bilayer can move freely between the phospholipid molecules is what we're looking for. The diagram shows a plant cell. Now, when a plant cell is placed in a water potential that's less negative than the cell contents, what happens? When it's less negative, this means it has a higher water potential. So when the water potential is higher, this means that water is going to move from a region of higher water potential to lower water potential down a water potential gradient via osmosis through the partially permeable membrane. Now, what is the answer? Is it C? Is it D? Is it B? Is it A? Okay, so what is shown in A is that nothing happens except the vacuole shrinks, which is quite abnormal for something where water is going to move in, so the volume of the vacuole should actually increase, which is what we see in C and what we see in D. In B, we see plasmolysis is happening, where the membranes are basically pulling apart from the cell wall because there's barely any water inside. This is definitely not what's gonna happen when water is literally moving into the cell. C or D. Now you can see that D, the cell wall basically is so flexible, the cell became twice its size magically. The cell wall is definitely not like this. It does not allow it to increase so much. However, C definitely makes sense. Being turgid is what's going to happen. The diagram shows a fluid mosaic model of membrane structures. So which molecules in the membrane are identical? So you have 2 and 3 or 4 or 1. Now you can see that 2 is definitely going to be different, and the branches of glycolipids and proteins are going to be different each time. 1 is a protein, and so the molecules in the membrane, the proteins in the membrane definitely do differ. However, cholesterol only has one structure, so cholesterol is going to be identical. The photomicrograph shows the stages of mitosis. Unfortunately, I do not have the pictures in this exam for some reason, so I won't be able to solve this question. So let's go ahead and move on. The diagram shows part of the organization of DNA molecule and associated histones. Which statement about the features labeled P, Q, R and during prophase of mitosis are correct? P, Q, and R. So we could see this is the DNA strand wrapping around histone proteins. There's a histone protein R, which is going to hold in place the cluster of histone surrounded by DNA. And let's see what are the options here. So the coiled DNA molecule forms Q and wraps around the histones of R to form small clusters held in place by histone P. So what he says here is that the coiled DNA Q, so we have Q, right? This Q, okay, it's the DNA. And then he says that histones R are going to be wrapped, so DNA is going to be wrapped around histone R, which actually doesn't make sense because it looks more like it's wrapped around histone P. And he says that it's going to be held in place by histone P, but I see that R is actually the one that's holding them in place. So we can just erase this and we can cancel out A. Okay, the groups of histones P and associated DNA Q move closer as the chromosomes condense around R. There's no way that we can move that they move closer and condense around R. Okay, because we do know that in prophase DNA does condense. However, we don't really know the way in which it does so. The histones P and R are made of proteins around which the DNA molecule Q is wrapped, and so DNA can fit inside the nucleus. I think that we can immediately cancel out C because nucleus is not even an option here. Prophase is basically the stage in which nucleus is broken down. We're not trying to fit inside the nucleus, we're trying to actually break the nucleus and break free. The linked groups of histones P and R in the associated DNA Q form strands that fold and twist together to form a chromatid. I think that makes the most sense. As I mentioned, P is the histone, which what's it called, and R is a histone, and DNA is wrapped around it and then form strands that fold and twist together to form chromatid. There's no specific way of folding, we don't really know, it's just basically fold and twist together to form chromatid. Bacterium E. coli divide once every 50 minutes at 36, and E. coli are grown on a medium containing only heavy nitrogen and 15 until all bacterial DNA contained heavy nitrogen. Some bacteria were moved from a heavy nitrogen medium and cultured in a medium with only light nitrogen and 14. Some bacteria were then collected after each 3 generations, and their DNA was extracted and centrifuged. Hybrid DNA contains heavy and light nitrogen. The diagram shows the possible positions upper, middle, and lower of the bands of DNA. The actual positions of the bands in the first 2 samples are shown, and then it's up to you to figure out where the DNA will be in the last 2 samples. Which proportion of DNA of the sample in 250 minutes will be at the upper position? Okay, so let's understand how this works first of all. So the first generation was made only of what's it called, heavy DNA. So let's imagine this over here is heavy DNA. And what's it called, this is basically a double-stranded DNA molecule from the first generation. Second generation, we added light nitrogen, so we added N15. So what happened was basically this DNA is going to semi-conservative replication is going to happen, and so each strand is going to come here and it's going to be replicated using the light nitrogen nucleotides, and we're going to have a strand that's going to be heavy, a strand that's going to be light. So something like this is going to happen. So the second generation is officially hybrid, and this is actually something that they mentioned here, if I remember correctly. So the second generation is going to be hybrid. And this is why everything here is in the middle, because 100% of the DNA is hybrid, and so all the DNA lines up in the middle because now it's half the initial weight that it was. Now, this third generation, what happens is the same thing, semi-conservative DNA application, where each strand is going to be copied using light nitrogen. So this strand is also going to be copied using light nitrogen, this strand will be copied using light nitrogen, and the last strand is going to be copied using, again, light nitrogen. So now you could actually see that 50% is hybrid and 50% is made of light nitrogen. And again, light nitrogen weighs obviously lighter than, is going to obviously be lighter than hybrid DNA. So light nitrogen should come at the top strand, and then what's it called, heavy nitrogen is going to be in the middle. So 50% of your DNA is going to be here, and then 50% of the other DNA is going to be here. Here we're going to have the hybrid, and then here we're going to have light DNA. So you could see that we had initially 100%, it went to 50%. Now what happens in the last generation is the following. So this guy is going to be replicated in this guy over here, and this guy, and then here, and there. And lastly, so you could see that out of 1, 2, 3, 4, 5, 6, 7, 8 of DNA molecules, we have only 2 of them which are hybrid. So 2 out of 8 is going to be 25%. Okay, so obviously if you multiply by 100, it's going to be 25%. So 25% of your DNA is going to be hybrid, only 25% is going to be here, and then 75% of DNA is going to be made of light nitrogen. So what proportion will be at the upper position? 75%. Okay, now I understand this question may be a little tricky, but a little bit of drawing could help you along the way, and actually you get used to such questions as you solve more past papers. So let's move on now. Different tissues and plants were supplied with radioactively labeled substance to identify which tissues were actively synthesizing mRNA. Which radioactively labeled substance would be most suitable for the experiment? Adenine, ribose, inorganic phosphate, and uracil. Okay, so to specifically identify that it's mRNA, we need things that are specific to RNA, again, versus for example DNA. So we know that adenine is found in both RNA and DNA, so we can cancel this. Ribose, inorganic phosphate, or uracil. Inorganic phosphate is basically found in both DNA and RNA. However, ribose is specific to RNA, and so is uracil. So 2 and 4 are the only answers here. Electron micrographs may show large numbers of ribosomes forming chains along mRNA molecules. What is the advantage of this arrangement compared to when ribosomes appear singly on the mRNA? So basically you have a one polypeptide chain, and then you have a bunch of ribosomes on them. So I feel like this is going to help make the polypeptide a bit maybe faster, because it's being translated from a bunch of positions at the same time, and then they're going to be connected together. So this makes maybe go faster. Let's see the options. Different polypeptides can be produced simultaneously. They're definitely not different since it's just one mRNA and a bunch of ribosomes on it, so it should be the same. Okay, fewer tRNA molecules are required to translate the polypeptide. There's the same number of tRNA molecules, there's the same number of mRNA codons. I don't see why would there be needed fewer tRNA. Large polypeptide chains can be produced. It's the same size of polypeptide chain, it's just a bunch of ribosomes producing the same polypeptide chain. The polypeptide chain can be produced more rapidly. This is what we have just said. Okay, which row is correct for adenine? Has a single ring structure, is a purine, it joins to its complementary base with 3 hydrogen bonds. Okay, adenine is a purine, and so it has a double ring structure. So the first thing is wrong. It is a purine, and it actually joins to thymine with 2 bonds only. So it's actually C. Okay. The graph shows the loss of mass in a pollinated plant due to transpiration. So what would have occurred at point X? So you can see that at point X, there's a sudden increase in the rate of transpiration because there's a sudden increase in the loss of mass. So what's it called? What could have occurred at point X is something that would have increased the rate of transpiration. So a plastic bag goes wrapped around the plant, is going to decrease the rate of transpiration. If you were to put petroleum jelly on the lower epidermis, you will decrease the rate of transpiration. The plant was placed in a dark cupboard, decreases the rate of transpiration because stomata are going to close. Warm moving air was blown over the plant is what you're looking for, because obviously blowing air is going to increase the rate of diffusion of the water vapor. Which row is correct for phloem sieve tube elements? Cell surface membrane, cytoplasm, nucleus, ATS, ribosomes. Obviously, for phloem sieve tube element, what we know about them is they have a perforated cell membrane, a cell wall, and they do have a cell surface membrane. They don't have a nucleus, and they do have, tiny organelles. However, they're not gonna have, ribosomes. They're just gonna have, a few organelles, like maybe, as we mentioned, cell surface membrane, a little bit of cytoplasm, no nucleus. So this is gonna be C. Okay, C, not D. Okay, C. An aphid such as a green fly uses a tubular mouthpart called a stylet to feed on nutrients in the plant, and it inserts a stylet through the surface of a stem or leaf. When a stylet is cut near the aphid's head, leaving the stylet in the plant, a liquid with lower water potential continues to flow out the plant. So again, it feeds on nutrients in the plant, so it should feed from nutrients from the phloem, which means basically this liquid that comes out is going to be the assimilate. Which statement explains this? The stylet in the phloem sieve tube element in which there is a higher concentration of solutes. That makes sense. The stylet is a phloem sieve tube element where there is a lower concentration of solutes. No, the stylet does have, the phloem sieve tube element has a higher concentration of solutes, versus the stylet is in a xylem vessel in which there is a higher concentration of solutes. Actually, no, the xylem vessel has a low concentration of solutes, and the stylet is not in the xylem to begin with. So the answer is A. Okay. Which statements correctly describe the transport pathways in a dicotyledonous plant? Okay, in a aplast pathway, water may move through plasmodesmata, and the symplast pathway, water may move through intercellular spaces. The aplast pathway may be blocked by the Casparian strip, and the symplast pathway may be blocked by the tonoplast. Let's explain what you have to know about the aplast and symplast pathway real quick here. So the aplast pathway is known as the dead pathway. So it's also known as the dead pathway. And it's basically supposed to be made of, non-living materials. So things like the cell wall, the intercellular spaces, all these things that are non-living are going to be forming part of the aplast pathway. And what we mean by pathway is basically it provides this road or, path for what's called water and nutrients to pass, like what's it called, to pass through. Okay, and so what's it called? Most water actually prefers to pass through the aplast pathway. We're talking about 90% of water prefers to pass through the aplast pathway, through the cell walls, through the intercellular spaces, because there's not as much resistance as compared to like the symplast pathway, which is the exact opposite. It's the living pathway. Okay, so the symplast pathway is made of living stuff, so things like the cell membrane, the cytoplasm, the plasmodesmata, okay, the tonoplast, so plasmodesmata, and so on and so forth. Okay, and obviously only 10% of water would prefer to pass through this pathway. Now, as is written here, yes, the aplast pathway could be blocked sometimes in the endodermis by the Casparian strip, which is basically this waxy substance that's going to be found in endodermis in order to regulate the flow of water. So the aplast pathway may be blocked by the Casparian strip. It's C. Let's see the other options. An aplast pathway, water may move through plasmodesmata, wrong. Symplast pathway, intercellular spaces, wrong. Symplast pathway, tonoplast, okay, no. Okay, the graph shows pressure changes during a cardiac cycle. So you have which row correctly identifies X, Y, X, W, X, Y, and Z. W. Atrioventricular valves close. So at W, we're just starting ventricular systole, ending atrial, atrial systole. So what happens is the atrioventricular valves are going to close, or the semilunar valves are going to close. So first the atrioventricular valves are going to close, then the semilunar valves are going to close, and then Y it's going to be semilunar valves are going to open, and then Z the atrioventricular valves are going to open. So this should be A. Okay, what is systolic blood pressure? Systolic blood pressure is the maximum blood pressure in the arteries. That's C. Okay. When they remain at high altitudes, their blood cell counts increase in humans. What does this occur? To increase the Bohr effect. The Bohr effect is going to be the same as their blood cell count increases. To increase the diffusion gradient for oxygen in the lungs is what happens, and to maintain the transport of oxygen 2 and 3. Which comparisons of blood tissue and lymph are correct? Blood has a higher concentration of proteins in tissue fluid because larger proteins are too big to pass through endothelial pores into the tissue fluid. It's actually right. So plasma proteins will not be able to pass through into the tissue fluid because they're so big and they're not flexible. Lymph has a higher concentration of lymphocytes than tissues, as a high number of lymphocytes are located in the lymph nodes. That's definitely true. There's a higher concentration of lymphocytes in the lymph nodes than there are in the tissue fluid. Macrophages are too large to leave to the blood to enter tissue fluid, whereas neutrophils, which are smaller, can enter tissue fluid and pass to the limbs. That's a very huge misconception. No, macrophages and all other kinds of white blood cells can pass and leave the leave the blood to go into the tissue fluid because they are quite flexible, and so they're able to push themselves through the pores. So this is wrong. Okay, so it's just 1 and 2. The number of different tissues which occur in the wall of major blood vessels, which row correctly identifies the main tissues found in the layers of a wall of an artery: outer layer, middle layer, inner layer. Okay, so a number of different tissues occur in the walls of major blood vessels. So what's it called? What happens in the outer layer? So there's a bunch of collagen. In the middle layer, it should be either elastic or muscle tissue. There's no collagen. And then in the inner layer, there's going to be endothelium, no elastic, or a tissue that's only going to be endothelium. So it's A. Okay, again, another option, no photomicrographs. So we'll not be able to solve this question. An athlete who smokes. Smoking is out of your syllabus, so we can cancel this question. Some of the requirements for an efficient gas exchange system are a large surface area and a short diffusion distance. What's correct about how alveoli are adapted to meet these requirements? So how they have a large surface area, collagen and elastin allow alveoli to stretch. There's no collagen in the alveoli. Gases dissolve in a layer of liquid to speed up diffusion. Gases dissolve in a layer of liquid to speed up diffusion. Not going to increase surface area really. Many folded interconnected alveoli or sac-shaped alveoli formed by squamous cells. So these two could actually work. Yes, there's many folded interconnected alveoli versus short diffusion distance. Capillaries are next to the alveolus wall, or red blood cells are very close to the capillary walls. What we're looking for is an answer directly related to alveolus, not the red blood cells close to capillary walls. So C is your answer. Okay. The diagram shows 3 types of cells found in the human gas exchange system. So you have the goblet cell, the epithelial cell, and the squamous cell. However, here the question is talking about cancer and tar from cigarette smoke, so this question is cancelled. Some common antibiotics are listed. The action of each antibiotic is written here. So rifampicin inhibits RNA polymerase, streptomycin inhibits 70S ribosomes, neomycin inhibits DNA synthesis, ampicillin inhibits peptidoglycan synthesis. Which of the following will affect only the bacterial activity? So only bacteria, meaning not affecting eukaryotic cells. Eukaryotic cells and prokaryotic cells both have RNA polymerase, both have 70S ribosomes, both have DNA polymerase. What we don't have, however, is peptidoglycan cell walls. So obviously 4 is the only thing to affect DNA bacterial cells. What could cause an outbreak of malaria in a country after it had been eliminated? Mosquitoes becoming resistant to insecticides. Yes, definitely, an outbreak of malaria could be caused by an increase in the vectors. Migration of population due to war, definitely, because when there's a migration of population due to war, this means there'll be overcrowding. When there's overcrowding, there'll basically be a whole lots of they're not going to move into a very, very, good place with all the accommodations they need. So obviously an outbreak of malaria could definitely increase in this case. Okay, malarial parasites become resistant to quinine. Yes, so increasing resistance of malarial parasites is going to obviously lead to an outbreak of malaria. So 1, 2, and 3. What disease is caused by a bacterium and can spread by airborne droplets? Okay, by bacterium can spread by airborne droplets because many diseases can spread by airborne droplets. So measles and smallpox can spread by airborne droplets. However, they're not caused by bacterium, and cholera does not spread by airborne droplets. So tuberculosis is your answer. Smallpox has been eradicated, but malaria and cholera have not. Which statements correctly explain this? Cholera pathogens in the intestines are not destroyed by antibiotics. That is true. Cholera pathogens are very hard to reach in intestines, so they haven't been eradicated for this reason as well. Okay, plasmodium antigens change during the life cycle, and since they change, it's obviously going to be harder to eradicate them and produce vaccines for them compared to, for example, things like smallpox. Smallpox antigens remain stable. This is another reason. Vaccines only work against viruses is not true. It's 1, 2, and 3. Okay. When a person is given a vaccination, immunity to certain pathogens develops. Which of the effects of vaccination are correct? social effects of vaccination are correct. Production of antibodies to protect against future infections results in artificial active immunity. Stimulation of appropriate lymphocytes. I think all of these are going to be true. All of these are going to be the effects of vaccination. So 1, 2, and 3. Okay, what's the first response by the immune system to a pathogen? The first response is always phagocytosis. Okay, now this is the end of this video. I really hope this video was useful to you guys. Make sure to like and subscribe if it was, and to obviously check out the free notes I have available for you in the comments section again, and I'll be seeing you guys in the next one.